Type 2 diabetes is a multifactorial disease caused by a complex interplay between multiple genetic, epigenetic and environmental factors. Recent studies have identified approximately 40 genetic variants that show genome-wide associations with type 2 diabetes. However, there is still limited evidence for how these predispose to diabetes.
The aim of our study was to examine if the genetic variants associated with type 2 diabetes interact with the human epigenome, and if the genetic variants introduce or remove possible sites for DNA methylation. We could show that approximately half of the genetic variants associated with type 2 diabetes introduce or remove possible sites for DNA methylation to occur. We could also show that all of these genetic variants affect DNA methylation in human pancreatic islets, the tissue where insulin is produced and secreted. Changes in DNA methylation due to genetic variation was also associated with changes in gene expression, alternative splicing, and insulin levels in the human pancreatic islets. Our data clearly demonstrates that genetics and epigenetics interact, and that almost half of the genetic variants that increase the risk for type 2 diabetes affect the human epigenome, exemplified by changes in DNA methylation. It is not possible to change the genetic sequence. However, it is possible to change the degree of DNA methylation of the human epigenome. The next step will therefore be to test if we can reverse the risk for type 2 diabetes introduced by genetic variation by affecting the degree of DNA methylation in the affected positions.